Everyone can see. No.
Okay. Okay, great. Yeah, so my name is Brigitte Kieffer. I did all my research, I would say, here in an institute in France, Strasbourg, France, an institute called IGBMC. And I just left 6 months ago to go to McGill University, where I am the director of a mental health research center. So I'm kind of in between. My lab is half there, half here. So what I'm going to tell you is, well, maybe the message. I will try to show you how by using mice, we have an incredible tool to understand a neurobiological system. So let's start with this. Opiates, as you know, relieve pain and produce euphoria. So this will be all about the opioid system. We know that the brain is made of millions of neurons, that these neurons form 1 trillion of connections in highly organized networks. And we also know that each of these neurons carries thousands of receptors and ion channels on their surface. And basically, within this huge complexity, what we've been doing in the last 2 decades is trying to understand how do they do this and where do they do this. So that's basically going to be the theme of this presentation. And clearly what I'll try to show you is that the genetic approaches were absolutely instrumental for us to understand the system. But this is also true for cannabinoid systems, dopaminergic systems, serotonin, all the systems. So we could also use this presentation as an example of something you can do to understand a neurotransmitter system. So we'll have 3 little parts. So I have many slides. I mean, we can discuss, you can interrupt me, we can see 20 slides over 50 slides. It doesn't really matter. What is important is that, we talk about what you're interested in. So there will be a flavor for everyone because, okay, we'll have the first, a little bit historical part. So this will be the recreational part, so you can relax. And then we'll go to the how. So here I will show you how by inactivating the genes in mice, we have been able to understand the functions of mu opioid receptors, delta opioid receptors mostly, and also kappa opioid receptors by studying behavior in these knockout mice. So that's going to be the second part. And then the part 3 will be where. So here we'll have 2 kind of stories. One will be how can we refine the gene knockout approach in mice to understand where the receptors operate. And the second part here, from the gene to a traceable signal, will be a novel approach we have been developing a few years ago to try actually and see the receptors in vivo and track them. So this is going to be cellular imaging. So let's start with the historical part. So this is a reminder. I guess many of you know the story of the opioid system, but just to go fast. The morphine molecule was characterized 2 centuries ago as the most active ingredient of opium. Clearly, morphine produces its very potent analgesic and addictive activities by acting on the brain. And from there, chemists tried, since 2 centuries, to develop a compound that would be a potent analgesic, but would be devoid of side effects. And very shortly after, in 1998, the company Bayer, a German company, deacetylated the morphine molecule and put on the market the first non-addictive opioid compound. It was called heroin. Okay? Heroin was put on the market as to cure asthma at that time. And basically, clearly we had not solved the problem. But it took about 20 years for physicians, clinicians to realize that this molecule was even more addictive than morphine. And from there, see, this reaction is very simple to do. You can do it in your kitchen. And this is how heroin started to be produced worldwide and became a major health problem worldwide. Okay, so now, 40 years ago, several teams at the same time demonstrated that a radiolabeled opiate is able to bind in a saturable and reversible manner to brain membranes. So this was the definition of a binding site. And there was born the concept of a receptor. There should be something which looks like a receptor in the brain for this molecule. 2 years later, the first peptide that naturally bind the receptors were isolated. These were met and Lu and Kiefferlins. You've probably heard of them. So clearly we had not only receptors, but their natural peptide. And these formed the opioid system. And in the next years that followed, pharmacological and chemists worked very hard and discovered that this system is extremely important to reduce pain, control pain. This system is extremely important in controlling what you could call the hedonic tone, euphoria, dysphoria. It's also important in responding to stress and in controlling emotions. And there's many other actions which are more peripheral, like controlling respiratory depression, controlling the immune system, regulating GI transit, and so on. Altogether, if you want to think of a unitary way to the opioid system, and this is what we discussed just in the previous talk, this system exists in our brain to teach us what is beneficial for us, for our species survival, and is also important to help us cope with challenging situations. So that's the general context. Now, molecular properties, the genes encoding the peptide were cloned in the early 18s, so there's 3 genes. They encode many, many peptides, but all these peptides have a common N-terminal sequence, tyrosine glycylate Phi, and this is the opioid pharmacophore. That's what binds in an opioid receptor. So you know the beta-endorphin and enkephalins and dynorphin. This is the longest peptide. These are short peptides, and these are basic peptides. And the receptors have been cloned much later. The first receptor was cloned both by Chris and I independently at the same time. That's when we happened to meet each other, telling our own data and showing the same result, basically. And this was a delta receptor. And from there, it was easy to clone a mu, a kappa, and a 4th receptor gene that is homologous to the previous, to the opioid receptor genes, but is a different receptor. It doesn't bind opioid. Okay? So we have 3 receptors that are called mu, delta, and kappa, encoded by those 3 genes. And maybe Chris will talk about this later on, but there is affinity. So basically, each peptide is able to bind to each receptor. It's all a matter of concentration at the synapse. The affinity are variable for peptides to receptors. There's more or less selectivity, but altogether, they're all able to bind to these receptors. So, what do the receptors look like? Typically, these are called, they belong to the family of G protein-coupled receptors. These receptors show 7 transmembrane domains. They are classically schematized like this. You may know that there's 400 genes encoding these receptors in the human genome. So many of these receptors, you know, dopamine receptors, many serotonin receptors, are G protein-coupled receptors. In fact, in pharmacies, 50% of marketed drugs activate or inhibit G protein-coupled receptors. So these receptors are extremely important in healthcare, in developing new treatments, new drugs. So this is what it looks like. Now, it took another 20 years. So from the cloning of the genes in '92, it took 20 years to get the structure, to get these receptors produced in large amounts so they could crystallize, and then their structure could be solved by X-ray crystallography. So the mu and the delta structure were published by the Kobilka lab Brian Kobilka got a Nobel Prize in 2012 for structure of mammalian G protein coupled receptors. That's a very important field at the moment. It's scaling up. Now there's new G protein coupled receptors being crystallized and structure solved every day now. This is the direction of this field of pharmacology, very exciting. And these are the kappa and the oral receptors crystallized. They look alike. So I won't describe the structure, of course, but just tell you that in one characteristic and one feature is that their binding pockets is wide open to the outer space, which is not the case for adrenergic receptors, for example. And the other thing I would say is that these receptors were crystallized with an antagonist bound, so in an inactive form, which is, in a way, a quiet form for a receptor. We don't have yet the structures of active forms of these receptors. And just to mention, because I will come back on this point later, the mu receptor crystallizes as a dimer. Of course, crystallized molecules are not molecules in vivo, right? A molecule is in a membrane environment. But this provides support to the idea that these receptors may, in fact, in a cell or in a cell membrane, may dimerize. So I'll get back on this a little bit later. Clearly now, in this field, the next step will be to get a structure of the active receptor. And it will be complicated because there's not one active receptor. There's thousands of active receptors. This is what I want to tell you now, briefly. Long time ago, pharmacologists had realized, yes?
Sorry, can you how many years later will be active?
Sorry?
So how many years later will be active confirmation?
Oh, you're asking if we need another 20 years? No, I think this is underway. It's all a matter of finding a way to stabilize an active form. And actually, the active forms of those receptors are highly dynamic. They move all the time. So to be able to trap one of these conformations and have it crystallizing is a technical challenge. So Kobilka Labs and Ray Stevens' lab and these labs are trying to develop either ligands or antibodies or whatever that could actually trap an active form. But it's going to come. I mean, huge efforts is put into that. So in the next, I don't know, 2 years, 5 years, maybe next year, it's all underway. This is underway. But then you go into something complex because one active form will be crystallized, and then people will say, yeah, but then that's the active form for this, but not for that. So I'm getting to that, right, to this complexity. So a long time ago, 40 years ago, people had developed agonists that they classified as mu, delta, and kappa agonists, right? And then they realized that those compounds in vivo or in cells would produce different effects. So why would different mu agonists produce different effects? So their idea at that time, before gene cloning, was that, well, each effect would come from a different receptor, right? But we know this is totally wrong. Today, that's the view we have of those receptors. The agonist binding will bind into the active site, a little bit schematized, right? Then we'll recruit G proteins, and then G proteins will recruit effectors, and then the receptor will also recruit other non-G protein effectors. So there may be many effectors, and in the end, whichever effectors you will recruit, you will end up inhibiting the neuron. That's what an opioid receptor does. And any actually GI coupled receptor will do on a neuron. And you'll also have long-term effect through phosphorylation cascades. Now, what has appeared clear in the last years is that not only the receptor protein is important. What is important, in fact, in the end, is the complex between the agonist, the receptor, and its effector. And this is what is defining an active receptor. So now in the field of pharmacology and G protein coupled receptor, a new concept has emerged. Well, before I go there, this is just an article that Chris and I wrote with Amina Pradhan, one of our, what should I say, postdoc student, mentoree. For example, talking about complexes, if you put all the literature together, you will see that mu receptors liganded to morphine bind to this type of effectors, while the same receptor bound to DAMGO, which is a peptidic ligand, bind different effectors. So in the end, the responses you get will be different, although you have the same receptor. So this is what is called the ligand biased signaling. So basically what is now well accepted, and there's a lot of drug development which is now based on this principle. Two drugs acting at the same receptor may recruit distinct effector systems, which in turn will produce different physiological responses and different therapeutic behaviors. So this now can be used. You can target a receptor and try to distinguish adverse versus therapeutic activities. And in the case of opioid receptors, so this is just a summary of the literature, but we have evidence so that this may operate in vivo. This concept has been way strongly demonstrated in cellular system. You translate cells, you look at different effector systems. But in vivo is much more difficult to demonstrate. But in the case of both mu, delta, and kappa receptor, there are evidences that this mechanism operates in vivo. And actually, by choosing the ligand, you may elicit a different response. Well, don't go into the details. I mean, that's not necessary. I won't go into that. So that's basically the messages I wanted to provide you regarding these receptors and their molecular properties. Okay? So I don't know if there's any question at this point. It's all right. We go on. So now we go in vivo. How? So from the gene to behavior. So basically, I don't go into details here. We created knockout mice. You all know how this is done by homologous recombination. This technology exists since 20 years already, I think. Yeah, 89, 90, 15 years. So we've been creating those mice, and basically I will summarize data which are relevant. What?
So that's 25 years.
Well, I'm lost in decades, so 25.
We're just getting 25.
Oh, you know, a decade doesn't make a difference, right?
A long, long time ago.
So let's see what we learn from these new opioid receptor knockout mice. So these are all data. So how many years? 96? 18 years, right? Am I right?
Yeah.
Or 22? 18 years. So if you delete this new opioid receptor, so the first question was, how does morphine act? Morphine produces many, many activities. So are these activities mediated by mu, delta, and kappa receptors, or are they mediated by a single receptor? And basically, if you delete this receptor, you will see in a tail immersion test that morphine analgesia, which you can see here, you know, as latency of tail withdrawal from hot water. Morphine is, start again. So if you use this test, which measures morphine analgesia, you will see that the time that the animal needs to withdraw the tail from the hot water will increase with time with increasing doses of morphine, right? So this is morphine analgesia in a control animal. But in the knockout mice, you inject morphine, but the animal will withdraw the tail as fast. Morphine has no effect. So morphine is not analgesic in those animals. It's a very simple demonstration that basically morphine mu receptors are the primary target for morphine effects. And this was then later demonstrated with many other morphine-like opiates that are used in the clinic. So you may know, so I told about heroin. You know methadone, which is used for substitution as a substitutive treatment for heroin addicts, buprenorphine, or fentanyl. You heard about all these compounds. All these compounds produce analgesic efficacy via mu receptors. You need the mu receptor for them to work. And we also tested then all the effects. So not only what we call the therapeutic effect, but what we would call the non-wanted or adverse effect, which is morphine reward, morphine dependence. I don't go into the details of the test here. Morphine-induced constipation, respiratory depression, immunosuppression. All this is totally absent in mice like mu receptors. So this was actually, this was clear cut. We did this with Raphael. That was our first collaboration. It was very nice. These old times. Easy. You could publish in Nature. It was really easy. Used to be easy. So we did this together. And this is something I like to say. This is a cross-disciplinary collaboration. So Raphael was a behavior scientist. I was a molecular biologist. I didn't understand anything in behavior. I was unable to set up an essay, understand, analyze data. And Raphael wanted to learn a little bit about molecular biology, didn't get it. So we just went together, and each one of us wanted to learn from the other. And from there, I mean, you'll see in the next, we have been doing lots of collaborative work that was extremely, extremely fruitful. And now you know a little bit about molecular biology. I know a little bit about behavior. So that's very nice. That's the way to work in science. So a little bit philosophical sentence on the way. So, so we have a single receptor target for all morphine effects. Good. I must say drug companies did not like it. Everyone would have loved to have analgesia for one receptor, addiction for the other receptor, so we would target one receptor and be safe. No, it doesn't work like this. So now, when those mice got a little bit more interesting, at least to my opinion at that time, was to offer them other drugs of abuse. So with Raphael, we sat one evening and said, why don't we give them other things? So I summarize here a lot of data. So I told you that morphine reward is gone. In fact, if you do, this is measured, can be measured by either self-administration experiment or condition-place preference. I can give details if you want, but basically, there's no condition-place preference, or it is strongly decreased in those animals. I told you this. Now, okay, this was done with George Cobb, actually. Now, you can offer these animals to drink alcohol. Alcohol do not bind to opioid receptors, but alcohol has many other molecular targets in neural circuits. But those animals that lack the new opioid receptors are not interested by alcohol. Self-administration and drinking would be strongly reduced. Then, with Raphael, exposed them to tetrahydrocannabinol. And if you use a CPP paradigm, you will see that those animals don't show a CPP or significant CPP to THC. Although we know that cannabinoid binds to CB1 receptor, not to the opioid receptor. And it's going to be the same thing with nicotine. So all in all, what is interesting, I'm missing something here. Okay, something has gone away. Let me go back. So the conclusion here, I'm missing a sentence here, is that basically rewarding properties of any drug of abuse is strongly decreased in those animals or almost absent. So we have a convergent molecular target for all drugs or most drugs of abuse. So it means that those drugs bind to their own receptor, and then you would have release of opioid peptides, endogenous release, that at some point will activate these new receptors that are necessary for the final reinforcement. So we then ask the question whether those animals would also show a modified behavior towards natural reward. Because in the end, why do we have these reward responses to drugs of abuse? You know, we are not born to consume drugs of abuse. There must be a normal role for these reward systems. And of course, these systems are here to teach us what is good for you. So what is good for your individual, for your species to survive. So mainly food, sexual activity, and social interactions. All these are important. Motivation to eat is slightly decreased in those animals, but what I'd like to show you here, which is getting increasingly important, is the experiment that Francesca D'Amato did long time ago. But what did she do? She asked me the animal to study maternal attachment. This was in 1999, and I thought, wow, why does she want to study this in those animals? So I provided her the animals, and it took 5 years, experiments, until she finally was able to demonstrate that those knockout pups, the babies that are 4 to 8 days old after birth, show strongly reduced maternal attachment. So that they wouldn't find their mother's nest, they wouldn't vocalize after separation from their mothers. So there was a total blunting of maternal attachment. So the interpretation at that time was that maternal behavior is rewarding normally, and this is extremely important to initiate bonding. But in those animals, well, maternal behavior is less rewarding because the reward response is deficient in the absence of neuroreceptors, then the bond wouldn't take place as efficiently. And at that time, she ended up her abstract saying that this may be reminiscent to autistic-like disorders. And in 2004, this was pretty provocative, and she had to fight to get her paper. Psychiatrists wouldn't like to hear about that. But then, recently, we decided to look at the adults. What happened now to these grown-up pups that have developed with a deficient reward system? So what I show you is a little bit amazing, because in fact we have been sharing these mice with many, many teams. We have been shipping these mice all over the world. Many teams have studied their responses to drugs, to pain, to painful stimuli, to stress, but no one has looked carefully for these particular behaviors. So just a little bit background: autistic spectrum disorders, the main hallmarks: social communication deficit, language deficit. This you can't study in mice, not yet. Soon we'll understand how they speak. Stereotypic behaviors. The classical theory is a cognitive theory and is basically based on the lack of empathy. But more recently came up the social motivation theory, and this theory posits that maybe deficient social reward deficit could be at the origin of autistic-like symptoms. And we have the perfect animals to test this, because obviously there's a deficient reward processing system. So Julie and Jérôme in my lab decided to look at these animals very closely. And they went through behavioral investigation, and they went through genetic and, like cellular molecular investigation. And then, so I will drive you a little bit through this data to provide you also with the idea that with mice, although these animals are pretty primitive for, I would say, human psychologists, there's a lot of things you can do with mice if you do behavior. So what did we test it? We tested social interaction, and basically that's what we call direct social interaction. You put two mice together, and you will see that you will count the nose contact, the following, when they follow each other, and the grooming, which is a self-centered behavior. And what you'll see is that the knockout mice show less nose contact, less followings, and more groomings. So the social interactions is decreased. Now we can do a little bit more sophisticated social interaction with either a familiar or a novel partner in a separate compartment that the mice can explore those animals. And what you'll see, I don't go into detail, is that a wild-type animal normally prefer exploring a novel mouse compared to a familiar mouse in this setup. But the mice lacking neuroreceptors don't make a difference between a familiar and a novel mouse. We looked at nesting behavior. Those mice are really bad at building nests. We looked at motor stereotypies. I don't go into details, but there's clearly much more grooming, which is the same as we saw here, but in a different equipment, I would say, set up. And they do a lot of circling. And also, we looked at what could be called behavioral flexibility in a Y maze, where these animals alternate across arms much less than the wild-type animals. So they show a kind of perseverative behavior. So basically, using those set of tests, we could show that those mice recapitulate what we call the core symptoms of autistic-like behaviors. So now let's go for secondary symptoms. This is anxiety. This is defensive anxiety, where the animal simply finds marbles in the cage and doesn't like them. It's a threatening object. So they bury marbles, and the mutant mice bury more marbles. And also, we use a novelty suppressed feeding test, where the animal has food in the middle of an intensely lit arena. So it's a conflicting situation that the animal would like to pick up the food, but it's a very anxiogenic environment. And in this case, you measure, just show you this, feeding latency, And the animals lacking mu receptors show a much longer latency to go and pick up the food. Okay, so that's increased conflict anxiety. We looked at motor coordination, clearly altered in the rotor rod and also in the string test. And we looked also at pace size. We looked at aggressivity using the resident intruder test. And so basically you isolate a male for a month, and then you bring an intruder in the cage, and you will measure the number of attacks and the episode of ptosis also. And what you see in the number of attacks is dramatically increased in the mu-knockout mice. These animals are highly aggressive. Sensitivity to seizures has been described as a secondary symptom of, for some autistic patients. And you can see here that responses to PDZ, sensitivity to a convulsive agent, is increased. So these animals show secondary symptoms also. Is this due to a maternal effect or not? We did cross-fostering experiments. So basically, the mu-knockout pups were raised by wild-type mother and vice versa, and we found the same phenotypes. So this is intrinsic to the mutation, not to maternal, not a maternal effect. Then we decided to test whether this mouse model would have some predictive validity. We just tested the risperidone, which is the only approved FDA drug at the moment to treat autism. And this risperidone is known to reduce some aspects of autism. And basically, I don't show you the data, it would take too much time. But what we showed is that chronic treatment with risperidone in these animals partially reverses those signs. This you can't treat, but this is just to show you. This is a table by Jacqueline Crowley, who is an expert on mouse behavior, mouse models. She wrote this book called What is Wrong with My Mice? Maybe some of you know it. And she listed all the tests you can do in mice to test anything reminiscent to autistic-like behaviors. And that's all the tests you could do. And just look at the green check here. Everything we tested in those mice, except the red, are positive. So actually, our mice score positive in most of the tests that we have used. Now we went a little bit into molecular aspects.
In terms of anxiety, why so different responses depending on the test?
Different responses?
Yeah, because you have anxiolytic or anxiogenic responses in the anxiety models.
Yeah. Well, we actually did the test in the plasmaze and as we did before, and we didn't find.
You didn't find anxiogenic, like?
No. So these are kind of more stressful situation, we guess, and this is why we got this phenotype. So I don't think it contradicts our data or negative or opposing results in plasmaze, but that's what we found. Yeah, we can discuss some, okay, we won't. So a number of genes have been associated in human genetic studies to autistic-like or to autism syndrome disorders, the spectrum of autism syndrome. And so what we did, neuroligin 1 and 2, for example, SHANK3 and so on, and actually animal models have been built for those genes. And here we tested the expression level of the transcript from each of these genes that is known to be associated to autism in humans, and we found that those genes are totally dysregulated in the striatum. Their expression is not normal. We then look at synapses in the striatum by electronic microscopy and found that there's more symmetrical synapses, right? And there's less PSD, so these are the inhibitory synapses, there are more. And here, the excitatory synapses show reduced postsynaptic densities. So we have hallmarks, anatomical hallmarks, which fit with the idea that a GABA-glutamate balance has been modified, as we know in humans. So, we went for a larger gene expression study in those animals. We looked at a lot of genes that belong to either GABA signaling or glutamate signaling, and these are a number of genes. So all the genes that are associated to glutamatergic signaling seem downregulated. So we decided to test whether we could use this model maybe to go for a new strategy for treatment. So is this animal useful to predict a new treatment? So this receptor, mGluR4, so it's a metabotropic glutamatergic receptor 4, is downregulated in striatum. This is just, so that's a receptor which is strongly expressed in striatum and is heavily decreased. So we thought, why don't we try to facilitate the activity of this receptor, which is now deficient in those mice, see whether you can reverse the symptoms. And it happened that there was a compound I call VU here, which is a positive allosteric modulator for this receptor, so it facilitates glutamate transmission via this receptor. And this compound had been developed as an anxiolytic. So we said, why don't we use it and try to help these animals? And basically, we treated these animals for 2 weeks, and we repeated a number of these tests. So I won't go through all of them, but I'll just show you here. So just look at social interactions, and we have the same kind of response in other tests. Vehicle-treated animals, for the nose contacts here, you see that as expected from naive animals, the mu knockout mice show lower number of nose contacts. If you now treat the animals, both wild type and knockout, with this compound, well, the compound has no effect on the wild type animal, but restores a normal number of nose contacts in the knockout animals. And you'll see the same thing here for followings, and you'll see for grooming also, you will restore the basal behavior, or the wild type, the control behavior. And that's the same for stereotypies, same for marble burring, same for the novelty suppressed feeding. So all in all, that's interesting because compared to risperidone in this model, we have a compound that is able to reverse deficits that have developed in those animals. And compared to the classical, well, the reference compound, we have no sedative side effects. Risperidone puts the animals to sleep. Actually, it's very difficult to make behavior with those. And it also improves social deficits, which risperidone doesn't do very well. So I conclude on this study. That's the only study I will show you in detail. So these animals show a broad autistic-like phenotype. I say after 18 years, I counted right here. So it's interesting to see that maybe that's a lesson for behavioral study. You have to, it's better if you have a hypothesis to screen and really design tests to test a hypothesis, because those behaviors were barely studied earlier. So we have a model with what is called phase validity. Yes?
Is there any polymorphism in the nuclear receptor in autistic people?
This has not been tested yet. We have looked with some, but not found anything. Okay. I contacted Thomas Bourgeron in Paris, who discovered the neuroligin 1 and 2, and who had cohorts of autistic patients. He had done genome-wide association studies. And I asked him to look in his cohorts, and he didn't find any link. But this is not surprising. You know that, yeah, no data means nothing. No result means no conclusion. And in addition, in mental health disease, the population, and particularly autistic patients, population are highly heterogeneous. And the causes are multigenic, of course. So getting a signal out of background is very, very difficult. So maybe it will happen one day. I mean, yeah.
And how is expression of nuclear in the cerebellum? Because it has been related with autism, and for instance, expression of mGluR4 was
Yeah.
quite high in the cerebellum.
There's no mu receptors in the cerebellum. So we think, so the stereotypies issues and the motor coordination issues are probably due to the striatal localization of the receptor.
Except in humans.
Yeah.
In humans, there is more receptors in the cerebellum.
So basically what we could do also is prove here that a deficient mu opioid receptor function or reward processing function may be a primary cause for autism. But you know, if you take cohorts of autistic patients, these guys have got a disease for multiple reasons. Only a subfraction of these people will show the autistic symptoms because of this reason, and maybe 1%. No, I don't say that this is the cause for autism. I just say that a reward deficient system may lead to a deficient social interactions, because a social interaction is normally rewarding, and in this case it is not, and then ultimately lead to some of the symptoms. That's what I say. Okay, let's be prudent, cautious, careful. Forget about this. Yeah.
Do you think that your whole phenotype could be initiated just by the fact that they don't deal with their mothers properly? Because you get the same effects, if I remember, when you do a maternal separation.
Yeah. No, because we did the cross-fostering experiment, so it's not the mother behavior.
No, it's the behavior of the new knockout to the mother.
Yeah,
the fact that they don't care whether it's
but then,
mother.
see, what has been shown is that if you block, so just to go into this, there have been other experiments, right? This is not new from scratch. So social interaction happens at the maternal stage, so at the pup stage, and then at the juvenile stage, adult stage, and at all stages, social interaction is rewarding. So if you're lacking this reward from peers, at every level, it's getting worse and worse. So here,
I think that in maternal, one of the main symptoms of maternal separation is depressive-like behavior. And at least in our early experiment, this animal has antidepressant-like behavior. So antidepressant, yes.
Because it doesn't care.
Yeah, but if you look, just go through the, I don't remember, forced swim or suspension test, you have a antidepressant-like behavior. You have a decrease in the despair behavior. So, and if it's just for maternal separation will promote despair behavior, will promote depressive-like behavior.
But doesn't paternal separation also cause some autistic-like symptoms?
Yeah.
Mm-hmm.
But
so there's different.
Yeah, but you have, let's say, maternal separation promotes autistic-like behavior, promote depressive-like behavior. Here you have like maternal separation, you don't have depressive-like behavior.
Right.
You do have as a result, there should be something else than maternal separation.
Mm-hmm. Yeah. Here, what is known for sure, if you go on, there has been an association with this polymorphism we're talking about, the one we talked about with John Carr, between this and what is called social hedonic capacity. So it's again the group of Francesca D'Amato, and she looked at social hedonic capacity in humans and found an association with this polymorphism and social hedonic capacity. So there is a link with in normal individuals and their way to enjoy social interactions. There's also pharmacological studies in rodents that show that receptor blockade, but this is basically naltrexone, so it's a general blockade, it won't tell you which receptor, but this clearly alters social play in juvenile rats. So there are other evidence indicating that you need an intact mu-opioid system to be able to enjoy and use and develop normal social bonding or affiliative behavior. Okay. Okay. So I'll finish with the yes.
This decrease in my first question is the M4 receptor. The expression pattern is more or less similar to the opioid receptor, like more enriched in the striatal patches versus the striatal matrix, and is the decrease in M4 taking place in
in those particular neurons. So I don't think things are as simple because the neuroreceptor is in patches. that could be pre and postsynaptic in patches. I can't tell, you know, difficult to say. We can talk about this later. And mGluR4 is in the D2 type neurons, so the indirect pathway, and is mainly presynaptic, so it's going to be downstream in the output of, you know, it's going to be one of the output projection from the striatum. So I don't think there's a direct effect between mu and mGluR4. I think mGluR4 is downstream, you know, 1 to 3 neurons downstream is affected. And so what we do when we use this compound, basically we compensate for a deficit. So during development, in the absence of mu receptors, these animals obviously have developed lower mGluR4 levels somewhere downstream. So we compensate by providing this positive allosteric modulator that will facilitate this, in a way compensating for the absence of mu receptor upstream. That's all. Thinking about it. I don't know if it's true, we didn't prove it. But it's not a direct interaction. Just 2 slides with delta. Delta receptor is not a mu receptor. It is very different. If you morphine self-administration is not changed, okay, so it doesn't mediate morphine reward. Alcohol administration, self-administration is increased, not decreased. Cannabinoid condition place preference is unchanged, and nicotine self-administration is decreased. So that may be the only similar response to the mu opioid receptor knockout mice. That's again all this collaboration with Raphael and with George Cook for the alcohol studies. But what was very important, and this is again all collaboration, Raphael, thank you. What about, why don't we look at responses of these animals in models for depression and anxiety? And this was back in the 2000s, so don't make me count, but it's a while ago. We decided we would look at the 3 mouse line, mu, delta, kappa, in the same test by the same experimenter. And what we found is only the delta knockout mice show in the light dark test box increased latency to first entry or decreased time spent in the intensely lit compartment, and this means increased levels of anxiety. And here in the 4th swim test, those animals would show longer immobility time. This means they don't fight as much. They show a more despair behavior, so what we say a depressive-like behavior. So this was a demonstration that delta, but not mu and not kappa, probably or certainly have an anxiolytic activity normally and an antidepressant activity in the normal animal. And this was very important because later on, when we discovered these companies that had developed delta agonists to treat pain, switched the compound in the neuroscience department to try and develop compounds to treat depression. And now there's delta agonists that are in clinical trials to treat depression based on this finding. And then many more findings later on that confirm that. This was especially for, where is she? You. No, she. You. Devant toi. You. We talk about impulsivity. Just wanted to talk about that. We talked about this. No, it's not a question. And just talking about addiction. As you know, I guess many people told you that, many previous speakers told you that, well, chronic exposure to drugs abuse undergoes many changes in the brain, and addiction develops for many reasons. And among these many changes, reward processes are altered, mood is lowered, so this is rather here, motivation is totally changed towards seeking the drug, habits have changed, and now habitual behavior has strengthened, and of course, and you heard this before, decision-making processes are disturbed, and inhibitory controls don't work as well, self-control. Now, if you look at the distribution of mu and delta receptors, so this is autoradiographic binding, I'll come back on this later, look at the expression in the cortex. It is very strong in the prefrontal cortex, which is actually extremely important for inhibitory control. So inhibit drug taking. So we asked the question whether mu and delta receptors could be involved in impulsivity. And I just mentioned this study because probably this is maybe the most difficult task you can do with mice, is because mice are very active animals, is asking those animals to refrain from doing something they are rewarded for. So basically this was developed by Cela in Kingston, Ontario, and she developed a signal no stop task. I won't go into detail, but basically you will train the animal to work for a reward. They will be trained, and then to get the reward, they will have to refrain from doing what they used to do. Okay, so that's the test. And in this test, efficiency ratios means, well, the higher you are, the better is your self-control, the better you're able to inhibit your behavior and no smoke when you should. Okay, you have a better control of your behavior. And in this test, she showed that mice lacking mu receptor are much better than control. I mean, these animals work like machines. You know, they no smoke each time they shoot, they refrain every time they shoot. And the delta knockout mice are very, very bad, very, very poor performers in this test. So this is interesting because it opens the activity of opioid receptors beyond mood and depression, beyond reward and beyond mood. And these receptors may actually be important for self-control and impulse control disorders. So that's a new area which may be explored in the future. So if I summarize what we have learned, and when I say we, it's not only us, it's many people in the field, both pharmacologists and mouse geneticists. And in particular, we haven't much worked on kappa receptors, but there's many teams like Charles Shafkin, you'll hear him tomorrow, that work on the kappa receptor. So the picture we have now is that if we think about reward, clearly high mu receptor activity means euphoria, the initiation of addictive behaviors. Whether neuroactivity is poor antidepressant, well, we can debate on that. A little bit more complicated. Delta is not very much involved in there. And kappa is clearly on the low side. Kappa activation is dysphoric. I didn't have time to go into that, but Charlie Shafkin will tell you all about this. Now, if we look at mood, clearly high delta receptor activity is anxiolytic and antidepressant, good for you, and kappa is at the other end. Okay, so that may be the way we can represent the differential of very different activities of mu, delta, and kappa receptors in the brain. So they belong to a system, but they have very different functions. So should I make a pause or I just go on? I go on? Yeah, okay, let's go on. So we go to the where. So first, refining gene knockout. So clearly now this is almost a prerequisite, but it's technically so difficult. Of course, we need to delete those receptors in specific brain areas and even better in specific neuronal population to know where the receptor operates to control these behaviors. And I'll show you simply 3 examples of knockout of these receptors that we made in at specific sites and show you briefly the data. This is just to give you a little bit of pain. The knockout of the regular constitutive knockout, total knockout of these receptors tells us that mu receptors are important for acute pain, much less so for chronic pain. Delta receptors are extremely important in reducing chronic pain, and kappa receptors are important for specific type of pain, such as visceral pain. So this has been mainly confirming what the pharmacology had told us before, right? Now, the idea is now to know where do the receptors kill pain. The pain stimulus is starting from the periphery, from the skin, or organs in the body, And the signal is conveyed through a primary step, which is in dorsal root ganglia, which is not in the central nervous system, which is peripheral system. And then it enters the central nervous system, and there's many levels for integrating the signal up to the cortex. Okay? So we ask the question whether, so you have opioid receptors, mu, delta, and kappa at all levels of nociceptive processing. So where are the important receptors? Are they here in the periphery? Are they central? And why is this important? Because if peripheral receptors are important, it means you can develop a drug that doesn't cross the blood-brain barrier and will not have central side effects, right? You can be fairly specific. If the main receptors are central, then you can't avoid, developing central compounds. So, well, I simplify a little bit, right? But so the idea was to develop a knockout for delta and mu receptors here, in those nociceptors, primary nociceptors. So the first cell that senses the noxious signal. So I just show you the data with delta. So we create floxed animal. We breed this animal with cray mice that express cray recombinase specifically in these nociceptors. I guess you're familiar with the technique. Yeah. And then what you see is that in dorsal root ganglia cell, small cells show much less receptors. So we clearly have deleted receptors in nociceptors, which are also small cells. Again, I simplify. And then if we look at pain, so the delta receptor is inefficient in responses to acute pain, right? So we have to use chronic pain models like CFA, inflammatory pain, or SNL, sciatic nerve ligation, which is a neuropathic pain model. If you produce inflammation in the paw, what you will see in wild-type animal is that these animals will show mechanical allodynia. That is, you go with a mechanical stimulus and you look at the withdrawal from the stimulus, and they will develop this pain response over days, and then this will go back to normal after 21 days. Well, in animals that lack the delta receptors only in primary nociceptors, you'll see that the pain will develop harder with more intensity and last longer. So this tells you that this population of peripheral receptors is essential for normally reducing this inflammatory pain. Now I go to, I comment just inflammatory pain. It's the same for the neuropathic pain model. Now let's use this compound called SNC80, which is a classical delta agonist. So this compound will specifically in vivo activate delta receptors. Okay? So if you now inject the CFA, produce inflammation in the paw, pain develops here, and you can see, so this is basal response. This is the animal that has received CFA and is in pain. So you see the sensitivity is much stronger. It will withdraw the paw much faster, And now if you inject the SNC80, you will see that gradually with higher doses, you go back to the normal response. So the SNC80 is analgesic, it kills pain. And if you do the same experiment in those mice that lack the receptors, solely nociceptors, you see that the SNC80 has no effect. It does not work. It's not analgesic anymore. So what we show here is now that these receptors, this particular subpopulation of receptors in this particular neuron is actually essential to reduce chronic pain and mediate the analgesic effect of the compound. So that's a way to show where things happen. It doesn't exclude that things happen elsewhere, but at least at that place, in those neurons, these receptors are critical. So the consequence is that you may be able, one consequence or one implication, you may be able to develop a delta agonist that is peripheral, doesn't cross the blood-brain barrier. It should be efficient. And this is being tested. I show you a second example of a conditional knockout. This is now we have the mu fluxed animals. So we have created a conditional allele for the mu opioid receptor gene. And we have bred these animals with DLX3 mice which delete what queer recombinase is expressed specifically in GABAergic neurons and only in the forebrain. Okay? So, you're not used to these pictures, but look here. This is a control animal, and what you see here is a sagittal section which shows you mu opioid receptor binding. Okay? So red means very intense, and then towards blue means low intensity. So what you see here is that the classical view of a mu opioid receptor with patches here in the striatum, very strong expression, and very strong expression in the thalamus and the habenula. In those animals, what is very interesting is that we see nothing in the striatum, it's all gone. But at the level here of the thalamus, the receptor is intact. So I don't go into the detail, but basically, this is the brain, a scheme. You have mu receptors everywhere, almost everywhere. And clearly, what happened is everything which is backward here is intact, and here is gone, particularly in the striatum. And you have heard during this workshop a lot of, I guess, speakers talking about VTA striatum, you know, these dopaminergic circuits which, you know, auto-regulates itself and which is extremely important to mediate reward. Well, in this case, we have an interesting animal because we're missing the receptors here and we're having still these receptors intact. And as John Carr told you just before, these receptors, mu receptors in the VTA, are supposed to be extremely important to disinhibit dopamine and be responsible for drug reward. Yeah?
Did you check that how they behave in the temperature tolerance?
Did we check what?
Temperature tolerance. When they be in the cold temperature, in the low temperature, and they are in the, for example, in the high temperature. Could they tolerate with this temperature or not?
You mean housing the animal?
Because here in the knockdown,
Yeah.
We can see the most of the receptors they express in the columns.
Yeah.
And it's very close to the temperature regulator core.
Okay. I don't know. So you mean measuring how the animal behave in a warm or hot temperature?
Exactly.
Actually, we never did that. I don't know. I can't tell you.
And also about their blood pressure.
Yeah, no idea. We didn't measure it. Should be done. What we measure, we measured morphine analgesia in those animals. It is normal. It works normally. So this is consistent with the idea that there are important sites for analgesia, you know, in hindbrain areas up to the thalamus. Now I'll just show you one piece of data. This is under progress, but let's expose these animals to alcohol. So you remember I told you that the mu knockout mice showed reduced alcohol consumption. And indeed, so this was done recently, we developed a continuous access, a two-bottle choice paradigm. So the animal is able to choose. He has two bottles, water, alcohol, and can choose. Voluntary consumes. And this is a continuous access, so the animal will consume about 4 grams per kilo per 24 hours. And what you see here is that the knockout mice consume less. That's what we expected. This is what we had published before. If we do the same experiment with this conditional knockout that still have receptors in the VTA, well, we also see a clear reduction. And now Sami, a postdoc in my lab, developed an intermittent access. So when the animal have access to the bottles, not all the time, but only at some times, then you increase their consumption. It's the classical bar effect. If the bar is open from 5 to 7 p.m., you'll run to the bar and you'll drink more. If you have alcohol all the time, well, you'll drink a little bit, right? So the bar effect worked. They drink more, and the knockout mice drink less than controls. And if you use the conditional knockout, well, they still drink less. So we have to explain that. This is amazing because, in fact, most studies have postulated that mu receptors in the VTA are essential for the consumption or the rewarding properties of drugs of abuse? Well, we have receptors in the VTA. Everything is normal. We just have deleted the receptors in the striatum. Still, we get a strongly reduction of the reinforcing properties. So now, I leave you with a question mark. We now have to study to delete receptors in the VTA. We have to go for electrophysiological studies and so on to explain this. But clearly it tells you that receptors in the VTA are not everything. It is much more complicated than that. Yes?
Oh, the most natural question would be what's the rewarding properties of morphine in those drugs?
Yeah. We're doing this. We're doing this. It's pretty different. We're doing this with Raphael. We've done this together. We're still banging our hand on the wall to try and understand. Yeah. It is not the same. So, and we should also test maybe cocaine or others. I mean, clearly this VTA striatum, circuit with new receptors at the two sides makes it complicated. So we'll see. We'll see. I think those approach tell us things about, the circuitry, which are different from what we know from global systemic approaches or global knockouts. Once you enter the circuits. So all in all, everything is a balance. If you use a systemic drug, you'll activate receptors everywhere, and you have some inhibitory receptors somewhere for a given behavior, activatory receptors everywhere for another behavior, and only for this behavior. And what you see is a balance from all these effects. So there's nothing more I can say at this stage. I don't know if I should do this very rapidly. Because this is relevant to addiction, so I'll comment this a little bit. You heard about recreational use. You know about the addiction cycle. Once you start binge consuming, then you enter this cycle where you go through a withdrawal state, and then you go through this craving state, and then you consume again. And the more you enter this vicious cycle, the more you consume for, I would say, not euphoria, but for compensated for the aversive states that develops, right? So in the long term, basically in protracted abstinence, what happens is that there's clearly lowered mood, and the true problem in drug abuse treatment, if I may say so, is to maintain abstinence. Or for an addict, I mean, the problem is only going to go to the hospital when clearly he cannot maintain protracted abstinence. And I should say that generally in the field, protracted abstinence, so after a long, long time, is very poorly studied, but it is very well known in the clinic that addicts, whichever drug, that are under protracted abstinence, have altered emotional responses. So we have recently developed a model of protracted abstinence in mice. Basically, we expose the animals to morphine in this case, a classical protocol that will make the animal highly dependent. And then we put those animals back in their home cage and leave them for weeks. And classically, what people look here is how the animal will consume drugs or respond to drugs. But what we looked at is different. We looked at emotional response. How do these evolve? And what Celia found is that after 4 weeks, not 1 week, but after 4 weeks, you can see a despair behavior that is getting clearly significant in a force field test. So despair behavior develops. And you can also see that social interactions diminish. So social withdrawal appears. So there is a dynamic plasticity during these 4 weeks that lead to the emergence of emotional deficits and social withdrawal in this model. What she also showed is that this behavior, you can prevent social withdrawal or despair behavior to appear if you treat the animal with fluoxetine, which is a SSRI, so a classical antidepressant, Prozac, right? If the animal eats Prozac during those 4 weeks of abstinence, they do not develop this behavior deficit. So this tells you that serotonin is involved. And she could show that only in the dorsal raphe, which is the nucleus of serotoninergic neurons of the brain, we have, after 4 weeks, a difference between abstinent animal and control animals. So there clearly is a serotoninergic mechanism which is involved in the long term. So we just wanted to test whether this dysregulation of serotoninergic neurons would appear because of morphine activity everywhere in the brain, or is it morphine activity specifically on neuroreceptors that are expressed in the dorsal raphe nuclei, right? There's very few receptors, if you look overall, in the dorsal raphe, but we need to know whether this regulation of the raphe is an indirect consequence of neuroreceptor acting in the accumbens of areas, or is a direct consequence of chronic activation of neuroreceptors in the raphe. And Pierre-Eric did this. So basically, you don't see well, but let's, yeah, you can't read. So this is now a heroin model, and basically what he did, he injected pre-virus in the raphe nucleus of fluxed mu mice. So basically what will happen is the virus will express query recombinase locally in the raphe nucleus, and since the gene allele is fluxed, the mu receptor will be knocked out or knocked down specifically in this structure. Okay? So what he did is he injected the virus or a control virus. One cohort of animal was used to measure receptor knockdown. A second cohort of animals was used to test naloxone withdrawal. We wanted to make sure that these animals had become dependent to heroin. And the third cohort of animals was used to measure social behaviors and despair behaviors. And what he found, first of all, if you use a GTP gamma S assay, I don't go into details, you can see that basically in the raphe nucleus, but nowhere else, we have a decrease of these receptors by 40%. So I would say that the knockdown worked very well. Now, this cohort tells you that naloxone withdrawal, the withdrawal score, is identical in control and dorsal raphe nucleus knockout. So it means that deleting, whether these receptors are here or not, doesn't matter. You'll have a general physical withdrawal, which is normal, right? So it doesn't happen here. Now, social interactions, if you look at this, so these are the controls where you see impaired social interactions, I showed you before, but with animals that have a deletion here, specifically in the raphe, there's no more significant difference in the social interaction. So those animals did not develop social withdrawal. And you can see this with both the, what is this? The duration of social behaviors and the grooming duration. Now, on the other hand, we looked at another behavior. Those animals show in the Y maze a deficit, actually in spontaneous alternation. I didn't show you this before, but just show you it here. And they also have this deficit, and this is likely due to neuroreceptors elsewhere in the brain. So what does this experiment tell you? It tells you that this neuroreceptor, of the chronic activation of this neuroreceptor, is essential to produce the social withdrawal of abstinent animals. Whereas these receptors at the level of the raphe nucleus do not contribute to other effects like physical dependence or working memory deficit. So that's another example of a local knockout that tells you something about where do the receptor operates for which behavior. Okay, so I'm finished with this, and I will end up with imaging. Is that all right? When should I finish? Fine? Okay, good.
I have a quick question.
Yes.
Have you seen autism or phenotype in what GABA mu knockout mice?
What is not tested yet?
But
We didn't do it. Well,
well, in terms like in the pups and mom interaction.
no, but we didn't do the experiment. I mean, you have to design the experiment, right? You don't see it.
weird behavior, the pup cycle and mother interaction is
Yeah.
impaired, that the pups would gain weight significantly less, and then you could kind of use it as a marker. I don't know.
Oh, the weight?
Yeah.
Because
They don't,
change.
no, there's no change, and actually what happens is with time, so the adult from age 4 or 6 weeks actually are heavier.
yeah, the social impairment, I think,
So-
when you know, it's only when you take the mother away, you know, most pups start screaming. These pups don't care, don't seem to care when the mother's away. It doesn't mean that they
The
have.
constitutive one.
Yeah.
What the bug was on cover.
Yeah.
Haven't tested it.
No, no.
No, no. You know, this behavior, you have to test them.
Yeah,
You don't see, I mean, these are very subtle changes. You don't see them if you look at the animals, right?
One question, do you know if there is a decrease of maternal care to the pups in this?
This I can't tell. I think Francesca did that. You know, in the 204 paper, I know she had the question and she made the experiment to answer that question, and the answer was no, but I'm not able to tell you which experiment she did. I forgot. Okay, but it's the answer is no. Yeah. No, so it's likely, but we have to do the experiment. That's difficult. It's likely in GABAergic interneurons within. So it's probably like in the in the VTA in substantia nigra where you have dopaminergic neurons. The idea is that the receptors are mainly on GABAergic interneurons, and that they may they should there inhibit the inhibitory tone. So these inhibit the neurons, and it seems in the dorsal raphe it could be the same thing. Yeah. So why don't we know where are these receptors on neurons? Because we have no good antibodies. It is very, very difficult to know on which neurons these receptors are expressed. And this is what I'm going to show you now, how we can do it. The antibody? Which antibody? From you? Yeah, but for the mu opioid receptor?
Yeah, there
Okay.
is one now.
So it's
There's
getting
one.
better, yeah. Mm-hmm.
But for a long
Great.
time, mine was the only one which worked.
Yeah, yeah, on tissues, right? Sections. Good.
We inject the AGBT in the dorsal raphe. It is later
Yes. Yeah. No, we micro punch. So what we do is we punch it's about the size of the raphe, a little bit bigger. And then we don't do membranes, we do homogenates because you don't have enough material. You do homogenates, and the GTP gamma SSA works on this material.
Then you say that you have like a 40%, but probably it's more because that is contamination from other population.
Yeah, I mean it's from this punch, so I don't know, from those neurons. I don't even know in which neurons the receptors are. So basically, it's not a cellular definition. I agree. It's just they are down. But they are done everywhere. It's a Cre virus in a fluxed animal. So everywhere you have Cre virus, you excise that piece of gene, and then where the gene is expressed, you see less receptors. But you get excision in all the cells, right? Okay. Yeah. This is a new tool, which now I think will be interested generally in the field of G protein coupled receptors and in molecular pharmacology. Let's put it that way. Classically, when you hear about reporter mice, these mice show fluorescence in cells. So a classical reporter mouse expresses a fluorescent protein under the control of a promoter, and basically within those cells, you'll have fluorescence filling up the cell. That's a classical. And you probably know about the rainbow mice and others. It's beautiful reporter mice available in many, many labs. But what I'm going to talk about is not a reporter for cell, it's a reporter for the receptor. So the idea was that we can't see the receptor. We can't even know in which cells are the receptors. So getting back to this antibody problem, and I guess you're going to talk about this, Chris, also tomorrow, maybe, I don't know, whatever.
Oh, I talked about antibodies as drugs.
Yeah. So, well, raising an antibody for a G protein coupled receptor in general is very difficult, except for the very abundant receptors like CD1. There's very good antibodies for CD1 receptors. But for opioid receptors, since many years, since ever, there were no antibody that would work on tissues, right? People were creating antibodies. They would work very well on transfected cells, where you overexpress cells. But on tissues where the level of protein is very low, there would be none. For mu receptor, actually, there was only one that Chris has made long time ago. There were 7 rabbits. One gave, this antibody, and this serum was like magical.
Gold.
But unfortunately, there's a limitation in that. When it's used in use, no one was able to do monoclonal antibodies. So basically, now there's one which seems to work. But after 20 and how many years? People have been trying since '92, right, to make these antibodies. So a few years ago, Greg, a student of mine, said, oh, I'd like to create animals where we would see the receptors. So we would do animals that would express a delta opiate receptor naturally fused to a GFP. And at that time, I told him, don't do that. Don't do that. It's way too risky. You'll never make it. You have a physics time. It's too risky. And he insisted very much. So that's a lesson for PhD students. He insisted like crazy. I said, do it. So it took him 4 years, right? Because it's 2 years to make a mouse, and then didn't work, and 2 years again. But finally, he was really, he was right. Sorry, he was really right, because this tool appeared to be absolutely wonderful. So the idea was, let's make an animal that would express this fluorescent receptor. Before we went to that, to the mouse, he tested in cell lines where this protein would work like a delta receptor. Would it bind ligands like the delta receptor? Would it signal like the delta receptor, internalize, and it looked good. So he made the animal. So I don't go into details, but that is a little bit more complicated than a knockout. It's a knock-in where basically you insert the GFP sequence at the right spot, upstream the stop codon, right? So basically, the engineering is that in the end, the animal will produce this fluorescent receptor in place of the native receptor. So I don't show you the data, but this receptor in this animal appeared to be fully functional. It works just like a normal receptor, and it is by definition expressed at the sites where the delta receptor is expressed, because it is replacing it. And it is also expressed at physiological levels. It is not overexpression. So what do we see? In fact, this tool is incredible because you can, by simple anatomical studies, you can learn a lot of things. And then you can do dynamics because receptors move, and you'll see this. They traffic, they internalize, they recycle. And we can do physiology. So let me briefly go through the 3 themes. What do we see? Well, this looks like green, okay. This looks like an autoradiogram. So the autoradiogram I showed you before, remember, are fairly fuzzy. Well, you can quantify, you have no cellular resolution, and it looks a little bit like this. You see strong fluorescence in the cortex, in the basolateral amygdala, it's very green. But what is nice with this tool, and you can't get with the autoradiogram, which is using a radiolabel ligand, by the way. So here, you can go to the confocal microscope and then get a cellular resolution. And this cell, for example, in the hippocampus, is not a green cell. it's a cell with green receptors, okay, which is different. And then you can zoom in, and then you can label, for example, this hippocampus with a GABA antibody, a GAD antibody. So in red, this is a GABAergic neuron, and these GABAergic neurons expresses, you see the green here, delta receptors at the surface. And you can do this in every brain area, and all in all, what you can do is you can revisit the delta receptor distribution throughout the nervous system. And there's still no good antibodies for the delta receptor, that's for sure. And this led Greg to a number of kind of controversial discoveries. It pushed the field a little bit because Greg was able to show that in fact these delta receptors in dorsal root ganglia are not co-localized with substance P. So I won't tell you the whole story, but there was this belief in the field that substance P and delta receptors were co-localized in dense core vesicles, and that basically when pain is produced, substance P would be released and would expose delta receptors to produce antinociception and kill the pain. It was a beautiful theory which fell down with this simple observation that these are not in the same neurons, so they can't work together. So that's a negative result, but that was pretty provocative at that time. And then for another example is that Greg was able to show, if you look again at the dorsal root ganglia, that mu and delta receptors are weakly co-localized. So they are mainly expressed on separate neurons. And this was interesting because it led him to show, with many other experiments, of course, functional experiments, that those receptors may actually reduce distinct pain modalities through different pathways, mu being more important in controlling heat pain and delta receptor more important in controlling mechanical pain. So this is just to say that the physical observation of receptor anatomy helps you providing hypotheses to test and revise a number of views. So we have kept on, and we have created animals that now express a mu receptor, which is fused to a red fluorescent protein, which is called monomeric cherry. So this again took 4 years, and then we looked at first images. Actually, it's very fuzzy. You don't see anything. No, I'm sorry for this. You know, it's not very good, but if you look at this neuron, we looked at these animals, we opened the brain, we looked at the neurons, we got scared because what we saw was everything inside. And it was extremely difficult, and you don't see it here, you would see it on my computer. A very thin line on the surface, very little fluorescence on the surface. Most is inside. And we thought, well, probably there's a problem. Maybe the fusion will aggregate receptors, they stay inside. We don't have the natural distribution. So I don't go into detail, but what well, first thing we did is use Chris's antibody. Yeah?
It's not that these receptors are present in the vesicle surface, and then whenever it's needed, they will be fused to this membrane?
Oh, wait, I'll go back to this later, okay? Yeah, receptor traffic, but that's just like a few minutes. So, yeah, where was I? Yeah, so we used Chris's antibody in heterozygous animals. So they have one allele expressing the normal mu receptor and another allele expressing this receptor. And basically, we overlaid, I don't show you the pictures here, the labeling with your antibody and the labeling mu cherry, and it's exactly the same pattern. So the distribution is normal. So then we said, okay, let's test morphine. So I don't go through all the data, but what we looked, morphine effects are purely normal in those animals. So if we do a GTP gamma S assay just to look at morphine signaling, it works perfectly well. If we do a morphine analgesia, hot plate test, tail withdrawal test, morphine analgesia is perfectly normal. Morphine condition, place preference is normal. All morphine responses are normal. So this is amazing because it shows us that only few molecules on the surface are sufficient to produce a full behavioral response. So this goes against what people thought. You know, it means that most receptors are inside, and there's only a few molecules there to react to the ligand. So when we looked at the brain, so there's no point I show you all this because it's not very good, but just to give you an idea, we find the striatal patches, of course, which is a hallmark of a mu receptor distribution in the striatum. I'd like to say here that it looks, basically, we find three types of signals. One is cell bodies, very clear. So this means that the receptor is expressed in the cell bodies, and at the surface, of course. So for example, in the septum, if you would see the original images, these are beautiful cell bodies, you know, nicely labeled. In the lateral hypothalamus, we have these giant cells which show beautiful cell bodies expressing mu receptors. In other areas, we see fibers, and in other areas, we see diffuse staining, like in the patches. And diffuse staining is difficult to interpret. You know, what does it mean? And we believe that these are presynaptic receptors on terminal. We still need to demonstrate it. And we believe that in patches, for example, there's some of the receptors, or a large part, is in terminals. Actually, to know this, we need to treat the animals with an agonist to internalize the receptors and move them back to their cell bodies. So then we would know. So we have to do those experiments. And the densest area, so this is a sagittal section, and this is just for information. You may have heard about the habenula in this meeting. Paul Kenny? No, he didn't give a talk.
Yeah, we did a medial habenula and EPD.
Yeah, okay. The habenula, I mean, is gaining interest in the field of addiction. That's an area which basically predicts aversive outcome. It's an epithalamic structure and highly conserved invertebrate. And guess what? This area is the area which expresses the most highest density of mu receptors in the brain. That's where you have the largest amount of mu receptors. And if you look on this sagittal section, you see this red track. This is the habenula interpeduncular nucleus tract. And if you, so now if you do a coronal section here at the level of habenula, you will see this. And now you can see the dense, these are mu receptor expressing cell in medial habenula. And if you go on, you can even see where, in which subregion of the medial habenula. And now if you cut here, you will see this extremely dense red fluorescence. And if you look closer, you'll see fibers, terminals, and cell body in the interpeduncular nucleus. So this is just to tell you basically that we can describe those, the distribution of those receptors throughout the brain. So we've been, of course, crossing these mice. So now we have animals that express green delta receptors, red mu receptors. And this is now a searchable brain atlas. You can go on the web and go through the brain and look at these receptors. And why did we do this? Because there's all this issue of receptor dimerization, I would say. That was the idea at the beginning. We know that the receptors functionally interact. What does it mean? We know that if we have a delta compound in vivo, the effect of the delta compound will modify the effect of a mu compound. You know, mu, delta, they seem to interact, but no one really understands how they could interact. Do they interact actually through circuit levels? I activate mu receptors, I release enkephalines, and they act on delta receptors and so on. Or do they interact within the same neuron because they are both GI coupled? So I activate one receptor, I recruit GI protein, so I impact on the second receptor that also recruits GI protein? Or do they actually dimerize on the membrane and interact that way? So this question has been discussed and debated since, I don't know, 10 years, and there's a lot of indication from transfected systems that the receptors can dimerize. Okay? It is possible, but does this happen in vivo? Well, there's a simple way, at least, to answer partly. Before you dimerize, well, maybe you should be expressed in the same neurons. So do these receptors are expressed at all in the same neuron? A very simple question. if they are expressed in the same neurons, they may dimerize or crosstalk by signaling pathway. If they are not, they basically, the interaction will be through circuits. So we did a complete mapping of neurons expressing mu and delta receptors in those double green-red mice. So I don't go through the detail. These are just some examples, but you don't see well, of neurons where the receptors are. Look at this neuron, for example. It is loaded with red. This is the green, and this is the double expressing, right, a neuron. And everywhere it's the same. You have the nice delta receptor on the surface and the mu receptors very much inside. But there's clearly a co-localization. And what we found, in a way, that was surprising. You have co-localization in separate neurons throughout the brain. However, in the forebrain, well, receptors are mainly in separate neurons, while in the hindbrain, receptors are mainly in the same neurons. So why is this interesting? Well, basically, opioid receptors are very much studied in mood control, reward, and cognition. For these high-order processes, it is likely that the receptors have evolved separate functions and are not co-expressed at all. Whereas for responses that involve very, let's say, survival mechanisms, like eating, sexual behavior, perception, and responses to aversive stimuli, for those, receptors are co-expressed in the same neurons. They may have evolved from those. Well, it's an evolution issue, maybe. The more the species have developed higher order function, the more receptors have acquired separate functions. Why is this important also? Because now, based on this co-localization database, there's a way to study heterodimerization in vivo, because now you can focus and isolate those particular neurons and circuits and study in vivo their potential dimerization. So that's anatomy. So that's all anatomy. So you can do a lot with a simple anatomical analysis of receptor expression within the nervous system. Now let's go for dynamics. So we said receptors move, right? It is known that receptors activated by an agonist will internalize, and then you have two types of, let's say, receptor behaviors, I would say. Either the receptor recycles back to the membrane, well, is basically dephosphorylated and then resensitized back to the membrane, or the receptor is committed towards degradation, or some of the two. I mean, but classically in the G protein-coupled receptor fields, receptors can be classified as fast recycling receptors and fast degrading receptors. There's two types. The delta receptor. Well, I don't know if I can do that. So this is, so now you cannot do see receptor trafficking in a live animal. The optic techniques are not good enough to be able to record within a living animal receptor trafficking. You need much higher resolution. It may come one day. So for now, if we want to see receptor trafficking, we need to use primary neurons. So basically, we take the neurons out of the mice, we plate them, and we can expose these neurons to the ligand. Let me see if I can have this movie working. Yeah. And see, this is what happens. Up. No. No. So it doesn't work. Yeah. So what you see. So that's the receptor moving in the cell. So in 20 minutes, so at the beginning, does it go back? No. Yeah. So at the beginning, the receptor is mainly on the surface, and then it sees the agonist that will bind. So it's a deltorphin peptide, and then the receptor will cluster at the surface, and then everything will go inside. So basically, after 20 minutes, this is 8 seconds, but it's 20 minutes contracted in 8 seconds. Everything is inside, and the receptor is not available anymore. And actually, if you test now the compound in the animal, and I will just show this to you, well, you'll see the animal does not respond to the drug after this has happened. So we can do this.
What was it? It was a very high concentration of the ligand?
Yeah, this was, so we tested from 10 nanomolar to 1 micromolar. And basically, you can see from 100 nanomolar, you can see the phenomenon starting.
Excuse me, is it a kind of, sorry,
What?
It was kind of prevention for the cell that not to be overstimulated. How you can explain it?
Oh, but this is a classic phenomenon in biology. You know, it's an homeostatic response. If you activate, you need to deactivate. And this is true for every receptor.
Right, but
right?
here is all of the receptors they are going.
Yeah, but see, it will be rescued. So let me go there,
Okay.
right? So if you now stain those neurons for lysosomes, which are the compartment for degradation, you will see that after 2 hours, most receptors are into lysosomes. So this means that you activate the receptor is internalized and goes towards degradation. So it's not very economical for the cell, right? One activation, receptors go to trash, basically. Okay, it's not always like that, but what happens, okay. Now, what are the consequences of this? Let's see what happens in vivo now, okay? And we'll get there. So this is a study by Amina. So basically, in vivo, what can we do? You know, we did put an optic fiber actually and a microscope in the head of the animals, but the resolution was not good enough. We couldn't see receptor internalization while the animal moved. But we can inject a drug, we can measure the behavior, and then we can cut the brain and see whether the receptor is like this. This means on the surface, it's a controlled situation, or like this, which means in vesicles inside the neuron. So you don't see it very well here, but once you're used to the picture, we can easily distinguish a neuron that has internalized receptors from a neuron that has normal or not internalized receptors. So what Amina did is she used 2 compounds. This one is the classical delta agonist. What it does is that, and this one is has been developed by AstraZeneca and is now getting used in the field. These compounds are almost identical. They bind to the receptor with the same affinity. They activate G protein with the same efficacy and potency. And they both produce analgesia and they both reduce anxiety at similar amounts, right, with equipotent activities. So they look very much the same, except one internalizes the receptor, as I just showed you, and the other does not. So Amina treated the animals. So if you treat the animal with SNC80, so that's the compound that induces internalization, what will happen is what you have seen. That is, if you look at the cells, receptors are all inside and are going to be degraded. You will produce analgesia, you will produce anxiolysis right away under the acute injection, and then you'll see the receptors being degraded. And if you look after 24 hours, you'll see the receptor back to the surface. So I'll say back, I don't know if this is right, they are at the surface. So likely the cell has restored a pool of receptors and that have trafficked to the surface. So after 24 hours, you get to the original situation.
But those which they went to the
I don't know if these are the same. I can't tell you whether this is recycling or whether this is neosynthesis, but it is likely that you have neosynthesis. Because from lysosome, I mean, it is only a small portion may be recycled. Most of it is degraded. So the cell has resynthesized receptor that is back to the surface, 24 hours. So now Amina asked the question, well, what happens if we treat, every 24 hours? Again, I come back with the agonist, I internalize the receptor. So she did this for 5 days. After 5 days, what she saw is that basically in the whole animal, you can't see the receptors anymore. So degradation was stronger and has overcome. the system, the cells were not able to restore enough of receptors. So with time, the receptor is degraded. So now, if she uses the agonist, she sees complete tolerance. The compound will be not analgesic, will be not anxiolytic. Of course, there's no more receptors, right? So that's a classical view of tolerance. Now, what is interesting is the other ligand. So now we have this RM, this compound, remember, produces analgesic and anxiolysis just as well as the other one. However, if you look at what happened after the acute injection, you'll see that the receptor is intact. It is on the surface. It has not internalized. Okay? So, now she injected the drug every 24 hours, exactly the way she did with the others. What does she see? Well, the receptor is intact. It looks perfectly well. It's on the surface. It's there. It's responsive. So now she injects the drugs and looks at behavior. What happened? You know, do I produce analgesia? No. I have tolerance to analgesia. Wow. Why is that? Do I produce anxiolysis? Yes, perfectly well. I can inject, and the anxiolytic effect is as strong. There's no tolerance. So we have two different effects. Two different ligands, almost identical, but the internalization effect is different. And now we have generalized tolerance versus analgesic specific tolerance. Yes?
Was this internalization process homogeneous in the whole brain? Because the most easy explanation will be in some area you do have internalization.
Everywhere, DRGs, perfect. No, we looked at every brain area,
was similar.
Peripheral, it was on the surface. Yes?
So, now the question which is coming to my mind is that the binding of the receptor and the ligand.
Yeah.
In the first case, I think it's like very strong binding, and this drug, this ligand must be destroyed also. And must be digested. So that's why cell taking all the receptor, the lysosome digesting the whole ligand and the receptor together.
Yeah.
But here, the binding of the receptor and ligand is not that much strong. So after a while, the ligand can be digested somehow, and then receptor is no need to go to the lysosome, and also it's gonna be digested and destroyed. So the receptor can come back to the surface.
No, that's not the explanation, because the two compounds are almost identical, and so first, I don't think they are.
They have a different in the structure.
Okay.
And it can make the difference in the binding.
So I don't know the binding. We didn't measure dissociation rates actually, but their binding property in terms of affinity are the same. I don't know. But I've got the explanation here.
What about the functionality? Because the fact that they are re-expressed doesn't mean that they are functional.
Okay. Yeah.
Maybe they are less coupled to GI or...
Well, let's say we have an explanation here which does not exclude other explanation. Right? I can't answer actually to this particular proposal because we didn't check at dissociation rates and at degradation of the compound, so I can't say how different these are. But what is different between the two is that producing analgesia and producing anxiolysis likely does not require the same signaling pathway. Okay, so we have a kind of biased situation. And so those different signaling pathways adapt to the chronic injection differently. And here, what we saw, I don't show you the data here, but the voltage dependent calcium channel that are coupled to the mu receptor, normally to produce analgesia, now are uncoupled. They are desensitized. So the receptor is still on the surface, but the effector is gone. So basically the cell has found a way to blunt the signal by cutting off the effector. And this did not happen in the brain and does not happen for the anxiolytic response, which use a different signaling mechanism. So that's our explanation. It may not be the only explanation, there may be others, but this may explain it actually. Okay, so that's typically an example of biased effect. So you have the same receptor. It's not the receptor, it is not different, it's the ligand. Okay? And this is a pretty dramatic, effect. Having tolerance for every effect or tolerance specific to pain is important and can be used actually when you develop a compound. This is the last. This is the last just for, so we use drugs. But does internalization happen, under endogenous release, under physiological situation? That's what normally should happen. And we have been trying to stress the mice, produce pain, scare them, whatever, throw them into water in the forced swim, trying to see internalization, because we thought, endogenous peptide release would be easy and, peptide would be released strongly and we would see internalization easily. No, that was very difficult. So in the end, what we did is we developed a paradigm, a behavioral paradigm, where we knew from the literature that there would be enkephalin release in the hippocampus. Okay, so we applied that paradigm, and that paradigm basically is what we call a context-induced withdrawal. So you give morphine, you administer morphine to the animal in a context every day, and then on day 6 you put the animal in the context where you used to receive morphine, and you don't give morphine, and what you see is a context-induced withdrawal. The animal shows withdrawal signs, and this is known to release enkephalin. So we used that paradigm and focused on hippocampus. And what we found is basically something like this. So it didn't look like a saline control, where you see the fluorescence on the surface, and it did not look like the drug-injected animal, where you see strong fluorescence in the, in vesicles. It looked like fuzzy, but inside, with still some receptor outside. So a half-half situation, and even different. And what was important is that we saw this only at a time 45 minutes, not 30 minutes, not 60 minutes. It was very transient. We saw this in only 9% of the neurons that express delta receptors, so it is very restricted. And it was very region-specific because there were other brain areas where we could see constitutive internalization, and others we saw none. Right? We could increase that type of neurons if we used an enkephalinase inhibitor. So this was clearly due to enkephalin release. And it always remained partial. We always had some receptors remaining on the surface. And then we used correlative electronic microscopy, and it's seen, but we have to work on this further, that the endocytic route is different. The receptors are not going in the same vesicles as do the drug-activated receptors. So what is the take-home message of this experiment? Without going into detail, the take-home message is, when you use a drug, you produce a massive internalization that is non-physiological, and that does what I showed you, kills all the receptors. They all go inside, they are degraded. It's a tough treatment. Under physiological situation, things are much more local, transient, and subtle. So there's no more economical issue here. You don't kill all the receptors. You subtly, locally produce internalization, and it is likely that there's always receptors remaining on the surface, so that you won't have this abrupt tolerance effect that we would see with the drug. So that would be my last message here. And I want to thank, so instead of putting, the pictures of all the collaborators from the world, that's what I put here. Here are team members. It's a non-exhaustive list, but all people that have contributed to the data. Some of the data I've shown you, all the senior people, the technicians, the postdocs and the PhD students, the collaborators all over the world. That's very important. So I mentioned collaboration with Raphael, this collaboration with Jan, Chris is collaborating since a long, long time. Jan Kitchen, George Koob, and others. Well, all these people. I showed you data from Francesca, from Sela here. and collaboration of the mice. I mean, we have been sending those mice, I think, in at least 60 labs around the world. So there have been many, many other studies that have been done with these animals. And I think we're okay. I stop here. So are you dead, half dead, or? Okay.
I was wondering if all this beautiful study can be also translated in rats, because I worked with Viviana, for example, in my PhD.
Yeah.
And what we found was that, for example, the infusion of the delta opioid receptor agonist in the nucleus accumbens of adolescent rats had no effect on social play. And here we saw that this receptor can have anxiolytic-like effect. And sometimes if a drug produces anxiolytic-like effects, can also increase, for example, social interaction.
Yeah.
So yeah, we just
so you found that delta agonists
no effect when infused in the nucleus accumbens. But of course, they were adolescent rats. It was just only one infusion. So was
and you expect something? So you looked at social play.
Yeah. Social play, we found that the infusion of Damgo increased social play behavior.
Ah, okay. And you did the same with a delta agonist?
Yeah, and also with the kappa agonist.
Yeah.
And with the kappa, we have the opposite effect. We have a reduction in social play.
But I would guess that if you do this in the amygdala, for example, you may produce a strong anxiolytic effect. The accumbens might not be the right spot to do this, which in the time of the test, I'm not sure it would be enough, would show you increased social play. But not sure.
Yeah, because actually in amygdala we perform with the endocannabinoids.
Okay.
And we found that there was an increase in social play after the infusion in amygdala of URB597.
But in those cases, you increase social play because you play on the reward system, I think. With the delta agonist, you will play on anxiety. And I don't know, at which speed or to which level you would modify the social play then. I don't know. It's an interesting experiment, yeah. Try the amygdala.
Yeah, so for your highly internalizing versus slowly internalizing agonists, was the time course the same? So the rate of tolerance, were they different?
No, well, we did the exact same experiment. Okay. So we didn't test whether it could be different. See what I mean? So we applied the same, injection every day for 5 days and tested the drug effect after 6 days.
Okay.
So I can't tell. In that paradigm, it was similar. What? Okay, whatever. Go ahead.
A question about the receptor knockout. So with the MMR4 agonist, did you actually change any of the other phenotypes that you measure in addition to the behavior? So did you rescue any of the striated gene expression?
No, but we looked at the C4 expression and immediate early gene expression in the accumbens, which are disrupted, which are modified in mu versus wild type, and we restored it. So there's also, a molecular change that goes in the direction to say that this is causally related.
Related to that, you also measure like the eye balance?
No, we didn't do. Well, we'll do this actually by imaging, by MRI. We have planned this experiment. We're looking at functional connectivity in those animals. So I know the answer within 3 months. Yeah.
Regarding the reported new opioid receptors, basically all of them are
they are inside, so I can't tell you whether they are internalized or not yet at the surface. If it's a pool that is inside, waiting to be externalized, or whether these are internalized. They are inside.
Something that has a similar data with the cellular receptor with the other neurons, with the cellular. Do you have any speculative speculation about what is the function of the receptor inside?
Inside? Well, it happens that, in fact, it is more clear people working in the G protein kappa receptor field develop similar tools or starting having antibodies available that a lot of receptors are actually inside. So the speculation could be, what is this, a reserve? Could be a reserve, so that whatever happens after activation of surface receptor, basically you have next pool is ready to come. So that could be one explanation. Now, I don't know if you need what should I say? A finalist explanation. Do they need to be inside? I don't know. Maybe.
Well,
No.
There is one thing that as soon as you kill an animal, you depolarize everything. You get an incredible dumping of opiates. As soon as the animal dies, we did microrevidalysis like 20 years ago, and we looked at as soon as the animal died, all the ATP just goes hot. So you depolarize every neuron, and you get this incredible dumping of everything which is in the neurons. This really kills the animal. So it's possible. I don't know if you've done, because you can't do lipids, right? So this is after you've killed the animal. So it's possible that the receptors are just going in because of rapid internalization, because of dumping of all the opiate peptide. It goes up like 100-fold endogenous opiate concentration. Not really doing.
Well, you mean in these animals, the receptor?
Because as soon as they die, because the potassium pump suddenly gone.
So it's better to give them some, for example, colchicine to stop any.
Well, colchicine screws up the whole system. But
let's say if we do primary neurons, from these animals, receptors are also inside mainly.
Yeah.
So
that
it's
should be okay.
I think this is their natural localization. Should they, what is the regulation of trafficking mechanism? The question is, because there are mechanisms that export, so there are exportation systems. So why, do you consider these exportation systems are inefficient, so they don't bring the receptor as much or as well at the surface, or maybe? Well, I don't know, I'm tired. I just don't know, it's amazing, but in the end, you don't need many receptors. The most surprising thing was that, 1% is outside, 99% is inside.
What happened with the internalization? Did you
Yeah,
also
so, yeah.
degradate it as the...
No, so, very different, very different. So what you saw with the green cells, you don't see with the mu. With the mu, what you'll see in the primary culture is that there's very few receptors on the surface. I don't have the images here, most inside. And you'll see internalization very transiently. In fact, these receptors recycle very, very fast. They are part of the other group of GPCRs. So if you look at 5 minutes, it's like you get lots of vesicles, and after 10 minutes, everything is back to normal. So basically, these receptors recycle very, very quickly. The dynamics is totally different. They don't go to degradation. And in animals, we have tried. So we have been injecting fentanyl, for example, which is an internalizing agonist, and then try, kill the animal, perfuse, cut, look. We see nothing. Because by the time we do all this, the receptor has recycled. So we are it is very difficult to see internalization images in the brain. And it's even more difficult technically because there's so much signal inside, it shadows in a way the weak signal we would see on the edge, on the surface. So it is technically extremely difficult to see internalization for this receptor because it is fast, it is weak in terms of intensity compared to what we do with the delta receptors. So it's a more difficult tool to study that.
So Brigitte, I'm intrigued by your studies on what you show that there is really a low level of the receptors at the surface, which you think are the functional ones.
Yeah.
How can you explain then the difference in efficacy, drug efficacy, with all these opiates, agonists?
Oh. This is a broad question.
I'm just not. I know it's a discussion, so maybe something to think about. Why then agonists differ in efficacy if only a little bit? I'm the surface is needed, you know.
Well, efficacy may
Or
not
even
be as
even
much linked, you know, to the amount of available receptor, but to the coupling and the signaling pathway. And I would more think about biased signaling rather than receptor density on the surface in this case.
Yeah.
You may have different efficacies linked to the different, ability of the agonist receptor complex to recruit, downstream signaling to produce whatever it has to produce. So I would rather think that distinct mu agonists would produce their different effects or different efficacies because they stabilize a distinct active form of the receptor, which then triggers, as I told you, in the biased signaling star, triggers a different response.
And you know what? There's some new data coming out that there's signaling from inside the cell as well.
Yeah, that's something.
Yeah, that's another thing. Yeah.
Inside the cell, which makes everything really complicated.
Yeah, but we want to keep to end up with a clear message, right?
There isn't a clear message.
Haha.
That's
nothing is black, nothing is white, everything is grey.
That's good. Gives us lots of grant money for that.
Yeah, you have a question.
You mentioned that in your autistic spectrum study, you had the decrease in the endorphin zodiac.
I have what?
Decrease in endorphin
For
receptors in the
decrease, yes.
Have you tested that which is like amygdala because there are some cell receptors in?
Yeah, there was no change.
So it was in the cerebellum?
Yeah, but there's very little receptors in the amygdala. Well, so we actually, for all these genes, we measured prefrontal cortex, amygdala, accumbens, and putamen. And yes?
Regarding to the bias signaling, what do you think that immunocytosis can affect in this?
Yeah, sure. I mean, of course. Well, inverse agonists, they all fit in there. You have a whole spectrum between a full agonist and a full inverse agonist, right? Agonist, partial agonist, neutral, whatever. There's no neutral compound actually. And then partial inverse agonist and full. All of these could, lead to different conformation and lead to different effects. It adds to the complexity. There have been less studies on biased signal. Well, you can't do biased signaling with blocking agent, right?
Well, you can because the joke. Charlie's going to talk about joke
Yeah.
activation.
Yeah.
Which is, it's not really an inverse agonist. He calls it a what's it called?
Well, he will say it tomorrow.
But basically the receptor can be turned off by a signaling cascade, by activating the joke, and he'll show you that, and it can last for 2 weeks.
So in principle, a partial agonist for one response would be an inverse agonist for another response.
Yeah.
So you could activate one signaling pathway and in addition block another signaling pathway. So that's
Pharmacology is getting incredibly
exciting. Interesting.
Yeah. I mean,
You can really play on this,
about it's becoming the most exciting area because there's so many different possibilities now. And reaction, as Brigitte showing, the activation of the same receptor, maybe a different receptor complex creates a different receptor complex, which can lead to different downstream effects.
So now there's screening strategies, with biosensor. Michel Bouvier in Montreal is developing this. So you can screen for compounds, for like 20 signaling responses, cyclase, blah, 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 whatever. And then 100 compounds for one receptor. You can test 100 different agonists on 20 different signaling pathways and get up with signaling profiles for each of them. And now you can try and, now the challenge is to link that particular profile with an in vivo effect. So then you can develop a drug that will activate that pathway with that efficiency, not the other pathway, and then you'll get the effect in vivo that you want.
But even that's naive, because every cell is going to have a different set of G-protein coupled receptors it can partner with, a different set of arrestins, and so even that is naive. So it's
and
really
you can
different.
dimerize.
Yeah, heterodimers.
So because one partner could be another receptor in the membrane, right?
Yeah, that's what I was going to talk about tomorrow a bit, about the heterogeneity. you know.
So he's going to
almost every
confuse
cell
you
is unique.
tomorrow. I try to be simplistic, and he's going to do the...
I won't try and confuse you. I'll try and say it's so rich, it's so beautiful and rich. You know, the pharmacology is very rich.
But because you are coming from the cannabinoid field, in the cannabinoid field, I
It's
see
always-
that inverse agonism is much more clear than in the opioid field.
yeah.
Yeah.
You know what? Because actually the opioid receptors, when you talk to crystallographers like Brian Kobilka, for example, those guys that try to crystallize the receptors. The opioid receptors are extremely difficult because they're very dynamic. They move like crazy, and they have many. No, the question is how many conformation, active conformation? 1, 2, 3, 10,000. I think opioid receptors are highly dynamic, and you have much more, I think, combinations than maybe other receptors. They have been difficult for this.
But actually, the delta receptor was one of the first to show constitutive activity. A long, long time ago, that was one of the first ones to show constitutive activity, delta receptor. But showing it in vivo and the significance of it, like cannabinoid, is very difficult. Actually, it's difficult to show the cannabinoid in vivo, right? Because, you know, if you block it, because you don't know if you've got an agonist which is working all the time. Because it's part of the lipid binder, right?
No, the cannabinoid field
It's a bit tricky.
But it's more easy to find the inverse agonism because you have a very low endocannabinoid tone. We were Rimonabang, you was working with. You have very, very clear inverse agonism, ex vivo and in vivo.
You may have less conformation. Maybe this receptor is not as, I say, fuzzy, you know? You may like clack, 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 free conformation instead of...
You never know if you don't, if you have
No.
an endogenous ligand which is working.
But activation makes it fun.
Yeah, I mean, it's just, everything's fuzzy. When it comes to this, it's just very difficult to figure it out. But I think it's good to talk about it because I think it's one of the most exciting things around is the heterogeneity in responses. I'm not scared of it.
No, but we have to think differently and we have to develop other methods.
Yeah, and you have to keep an open mind about different drugs at the same receptor. But you're not always going to get the same effect.
And this is also the reason why it is so important to go in vivo.
Yeah.
Because very often, you know, what you may think of or anticipate from transfected systems doesn't work in vivo. In vivo is much more complex, and this is why you get surprises. And all the tools we develop, the conditional knockouts or the knock-in mice, bring new data. You know, things we didn't think before. So the technology is important. I think the technology also drives the field. We need new methods to see new things. Okay, let's stop here. We need a break.